Hi everyone. This we'll just get started on this speaker series. And also just as a disclaimer, this is going to be recorded. So if you have any questions, please feel free to reach out at the end of this presentation. But anyway, hi everyone. Thank you so much for joining the Sanford Institute's second spring speaker series for the year. Before we get started, just know that this speaker series session will occur almost every month of 2026. So just stay tuned on Eventbrite, and the summer speaker series session at least will be published maybe sometime next month. But back to today's speaker, we have Dr. Zhu, who will be presenting on the dopamine pedal for feasting with friends. Dr. Zhu is a postdoctoral fellow in the UC San Diego Department of Neurosciences in Scott Sternson's lab. His research examines how neural circuits and neuromodulators link internal state and social context to motivated behaviors with a focus on hedonic eating and aggression. Supported by a Sanford fellowship, his current work investigates how others or observing others eat recruits specific brain circuits in mice to trigger socially cued eating, revealing a previously underappreciated social control of eating and motivation. His work has previously been published in Science and Neuron. Please feel free to submit any questions through the Zoom chat function during the presentation, but note that if you want these questions to be answered, they'll be answered at the very end. If you want your questions to be anonymous, you can also privately message me through Zoom, and I can answer or I can ask the question on your behalf. Without further ado, please welcome Dr. Zhu.
Okay. Yeah. Thank you so much for your kind introduction and for having me today. So my name is Zhenggang Zhu. I'm postdoc in Scott Stenson Lab at UC San Diego. Today I'm really excited to share my research. Today actually the title changed a little bit. Present my research about the needs, perspective on empathy and compassion. Okay. So what is the purpose of life? So for most of animals, actually life is organized around their needs and the motivated behaviors that used to fulfill the needs. So ideally, once those needs are met, life is in balance. So right now we are living in the world actually saturated with abundant primary rewards, for example, delicious food to fulfill our needs. However, things are not always getting better. In sometimes, some ways, actually, things are even worse. So, for example, we are living in the world with these rising epidemics of metabolic disease, obesity, diabetes, and also this increasing rate of these motivation deficits in social anxiety, depression, and also social autisms. So these crises highlight a critical gap in understanding of basic needs and motivations. So to understand the basic needs and motivations, it's important to appreciate these two important frameworks. First, like motivated behaviors, eating, drinking, or socializing are guided by different types of needs. So Maslow's hierarchy of needs provide a useful summary. From basic level, we have the physiological needs, food, water, sleep, and then we have the safety needs. And then beyond that are these social needs for belongings, love, esteem, and eventually for human beings for like these self-actualizations. And second, we need to appreciate actually these motivated behaviors are composed by different behavioral phases. So the seeking phase comes first, which involving like sensing what is needed and evaluation of the desired outcomes. So once this outcome is found, this is followed by the second phase, the consummatory phases of eating behaviors, involving like consummating the goal of the behavior, like eating food or like a social interaction with the others. And the satiety phases develop during the consummatory phases, eventually terminate the behaviors. So by incorporating these two important frameworks, understanding what is being needed, what behavior phases you're looking at your behaviors, will better understand the purpose of life and also eventually understand like how the biological intelligence is involved during these processes and also contributing to our understanding of empathy and compassion. So research in the past 2 decades actually identified these different neural circuits and the neurodynamics could mediate different phases of, for example, eating behaviors. So including our lab and other labs, we identified the neurons called HRP neurons in our hypothalamus. So these neurons can specifically sensing the need for food and specifically promote the food seeking phases, not the other phases of eating behaviors. And nowadays, these brain body signals can be easily mimicked by these like GLP-1 drugs. It can selectively promote the satiety phases. However, there are still some gaps. So today we're going to talk about like understand these basic needs and motivations could bring us the opportunities to understand empathy and compassion, and also what are these remaining gaps for addressing to understand the needs and motivations. So first, why I'm talking about these basic needs and motivations, this is actually because empathy and compassion can be certain forms of these needs and motivations. So it involves, for example, definitely involve like social processes, but also it could involve the other like needs. For example, from learning from the social information, we also know actually it can indicating the needs for safety, and learning from the others, we are also good indicating ourselves physiological needs. And also, this compassion and empathy is also composed of these different behavioral phases, including the seeking phases, consumptive phases, and the satiety phases. But compared to our self needs and motivations, so empathy and compassion can be more complex because it also involves the social context, right? So the framework basically for the empathy compassion can involve like sensing others' needs, both positive or negative, and like positive part of you can say like physiological needs, whether they get some reward, get some food, and the negative part can be say, or whether they have strong safety needs, they are facing dangers. And then by sensing the other needs and compare or evaluate as compared to our self needs, and then we will finally decide, okay, maybe we should interact with the others. So, for example, for human beings, we can even do like take a lot of actions to help the others to fulfill their needs, right? So by understand these basic frameworks of empathy and compassion, we can better understand how we the biologists. So that's why it's really important to understand the biology of the basic needs and motivations, and then to better understand our empathy and the compassions. Okay, and also there's actually many gaps of understanding like needs and motivations. For example, I will list some. So compared to like these physiological needs or social needs, actually the safety needs, especially aggressive behaviors or aggressive states for protecting the safety needs remain poorly understood, and also as compared to the seeking phase or satiety phases, the consumptive phases of motivated behaviors, like how the motivations being sustained, remain poorly understood, and also how the needs, motivations interact with each other, and are there any computation rules for encoding different types of needs remain poorly understood. So my research vision is by understanding these basic processes, and then eventually will help us to understand the empathy and the compassion in animals and also in human beings. And that vision has guided my research in the past actually 12 years. And today we're talking about how my published work like bridge the gap in understanding these first two questions, and then how my ongoing work, especially funded by our institute, the Sanford Institute, help us to understand the need interactions, the computation rules, especially how it related to empathy and compassion. Okay, so first part, during my PhD, I really interested to understand the aggressive state that protecting our safety needs. So not just me, actually, humans have always been interested in like aggressive states or anger. So for example, 2000 years ago, Greek philosopher Aristotle said, anyone can get angry. This is easy. But to do this to the right person, to the right extent, at the right time, and in the right way, that is not easy. Okay, actually, 200 years before him, Eastern philosopher Confucius have similar ideas. So both quotes show how remarkable for our brain or bodies can choose aggressive state. The mechanistic studies start from Darwin. So he actually noted different animals may have these evolutionarily conserved aggressive states. For example, like you can see the angry faces in his dogs, kids, and our human faces, right? And also Nobel laureate Dr. Hales developed tools called electrical stimulation. He found actually it's the brain. If you stimulate the brain, especially subcortical of the brain regions, you can trigger the aggressive behaviors. And another Nobel laureate, Timbergen, actually he clarified the aggressive behaviors as a certain types of survival behavior that protecting our basic needs. And the technology has evolved a lot since then. But a very basic question: what is the biological basis of aggressive state? Remain poorly understood. So actually, if we say any cells control this aggressive state, those cells should respond to threat and aggressiveness. Activated then should elicit general attacks, not just in males, but also should evoke attacks in females. And activating then should elicit not just the behavior, but also these autonomic activations. And inhibit them should suppress attacks. So 5 years ago, my research identified very mysterious region, like its name called posterior substantial innominata. Innominata means it's unnamed region. So we found these regions in the amygdala part could mediate fit meet these four important criteria. For example, we found these PSI neurons. It can scale with social threat and aggressiveness, and use of genetic method to selectively expressing these opsins in the surface of the cell. And then use light, we can turn on the cell. We found turn on these PSI cells can elicit these generalized attacks, and also not just in males, but also in females. And also activating these PSI could elicit these autonomic activations like these pupil dilations, rapid heartbeat, respirations, and inhibit these regions to suppress attacks. So to my best of knowledge, these first regions that meets all these four criteria in mammals. Okay, so right after we published the paper, so like Dr. Faulkner, a principal professor, listing our region, this PSI region, as a key node for the aggressive state processing. Okay, so but still a kind of confusion point is we do find activating PSI elicits similar aggressive state in males and females, but we know naturally many animals, different species of animals, they show very different aggressive state in male and females. Like in mice, actually, male mice are much more aggressive than female mice. So why is that? What is the logic there? I soon realized the logic is naturally the PSI could get more activated in males as compared to females. So following that logic, I map out, okay, what brain regions could control these regions. We do find actually this PSI actually receiving the aggression information directly from a region called VMHVL. This is the most aggression center, okay? And very interestingly, we found there's sexual dimorphic EI balance. What I mean, there's an EI balance difference. So it means actually the circuit from the VMHVL to the PSI. So they send both excitations and inhibitions to the PSI. But in males, there's strong excitations to activate the PSI. But in females, there's stronger inhibitions to suppress PSI. So in general, that leads to like PSI get more activated in male mice. And also not just input to the PSI, we found actually there's a molecule called HCN1, it's an ion channel in the PSI neurons. But further lead PSI get more activated in males. So this published research actually uncovers the circuit and also molecular mechanisms for this aggressive behavior, aggressive states. So this bridges the gap of understanding the biology of safety needs, especially the states that are protecting these safety needs. But actually, the regions, like this SI region, has been neglected for a long time, but it's not a negligible part of the brain. Like in mice, these regions has more neurons than this like well-known region like that hypothalamus, and also in humans, actually, it covers 100% of our clonal section of our brains. So right now, we are collaborating with psychiatrists to study the assess functions in human patients. So stay tuned. And then next, I ask another key question: what is the biology basis of like eating behaviors for even more basic needs like the need for food, right? So actually, the neural basis of hedonic eating was remain largely unclear. Hedonic eating refers to eating is driven by palatable taste, not by hunger. It can lead to overeating and eventually cause obesity. However, the mechanisms is poorly understood. For example, at least many concept has not been tested, has not been validated. For example, how does the food palatability could shapes this motivated behavior? What phases it controls, right? And also these satiety signals, which especially these GLP-1 drugs, how they modulate these hedonic eating behaviors, and what circular mechanisms could sustain these motivations. And because we know dopamine is well known for its role in motivations, what role does dopamine play in its hedonic eating behaviors? So, because of timing, I'll just briefly mention we found food palatability can selectively promote the consumptive phases of eating behavior, not the other phases. Okay, that also highlight, okay, it's really important to understand what behavior phases you are looking at of your behaviors, which has been neglected for a long time for the field. And the other thing we found, actually, these GLP-1 drugs can transiently suppress these palatability signals. Means there's a kind of appetite recovery during later phases. So right now we are testing this synergistic effect of targeting the GLP-1 system and also this palatability system to see whether it can further improve these anti-obesity therapies. And then we're talking about more about these later two questions. So first, what is the circuit mechanisms for sustaining our hedonic eating behavior? So several years ago, our lab identified very important node for control this sustained eating behavior. So we found actually eating will suppress this hindbrain region, these populations called perioccipital neurons. So eating will suppress it. The interesting part is the suppression will be even strengthened if food is more palatable. Okay, and of these them prolongs the eating behaviors. So this hindbrain neural population is a negative feedback node for sustained food palatability. And more importantly, we found we build direct connections with these palatability signals in these hindbrain regions with the dopamine. So we found the logic is the palatability will suppress these perioccipital neurons in the hindbrain. And then it will suppress these VTA, vegan neurons, these inhibitory neurons. So this double negative cannot cause the activations. So it will activate the VTA dopamine neurons and then eventually increase the dopamine release to the downstream of this VTA. So that builds a mechanistic link between its food palatability with the dopamine signals. So this is a new mechanism for controlling its palatability driven eating. Or potentially explains, okay, why can't we get stop eating if we have something really tasty? And the next question is, okay, how the dopamine involved. So dopamine identified by Dr. Carson is as well as we all know, actually, we think is a very important molecules for regulating motivations and usually released from midbrain, especially from VTA. But actually, classical study has already found VTA dopamine neurons can activate during the food cues, during the food approach. However, its functions of VTA dopamine neurons activity during these consumptive phases of eating behavior remain poorly understood. So here, actually, my published research filling the gap by finding this VTA dopamine is actually showing this persistent activity during the food consumption. And also, correlation doesn't mean causation. What is the causal laws for the consumptive behaviors? We actually found these VTA dopamine neurons, actually, the published research has been showing controversial roles in food consumption. So use epigenetic, hemogenetic, or pharmacology to increase dopamine signaling. So as you mentioned, all these manipulations have been mentioned in his published results. They didn't mimic the natural dynamics of VTA dopamine neurons in eating. They are not turned on just during the consumption phases. They either show no role or reduce food intake. So this controversy has not been resolved. So by to resolving this controversy, actually, I developed a new method called this palatability calibration activation of video dominance, which means I just turn on the video dominance during the food intake, during the food consumption, and also making sure the activation of video dominance just mimic this high palatability food intake, not overshoot the video dopamine activities. We found only this physiological relevant activation of video dominance that can prolong food consumption. Not the other, like at least random activation of video dominance doesn't have the loss. Okay, so that's the take-home message from this published paper suggesting, okay, for understanding motivated behaviors, especially combined with neuromodulation, neural recordings, timing is a key. Okay, so my these two work actually bridge the gap of understanding the basic like safety needs and also the consumptive behaviors of consumptive phases of eating behaviors. And now I was talking about this the other two directions, especially funded by Sanford Institute to understand the eating actions and also the computation rules of encoding different types of needs. And yeah, so first I want to mention the different type of needs. Actually, animals often facing multiple needs, not just one needs. And these needs often interact with each other. These motivations often interact with each other. So that's how biological intelligence evolved. And that also potentially explains how especially for like human beings, how the empathy and compassion evolved from these like mammals eventually to primates and to human beings. However, I want to point out that so current research mostly focusing on one need at a time, which, for example, in the social research, we usually focusing on direct social interaction. And for people study the physiology, we usually focusing on one's own physiology or like one's own feeding behaviors. So for these reasons, the biology of need interaction remain poorly understood. Okay, so to, to addressing that, actually, I propose a framework to understand the need interaction. So first, we should capture how multiple needs naturally they interact, and then we can develop models to incorporate these different type of needs, and then study how they interact with each other. And then we can study the behavior mechanism, the biological mechanisms through how one need affect the other need. Okay, so I was talking about how one example of using this framework to study the need interaction, especially social and food motivation. So actually, we know feeding often happens socially across different species. For example, in insects, birds, fishes, and different types of mammals. And also, for example, for humans, we often go to our favorite restaurant to celebrate our, like, for example, paper acceptance or nowadays, especially grand announcement, right? So then today we're going to talking about our mouse friend, how these like us, these two different motivations, social and food motivations, may interact with each other. Okay, so actually we know naturally there are reciprocal connections between social observations and eating behaviors. For example, you can often at San Diego you can often see actually seagulls would rapidly approach each other when one seems to find food, right? This is because seeing a member of your species getting food provide a lot of important information, including food availability and also potentially about the food quality. And for mammals, actually, the social observations is really important for eating behaviors during our interaction with our parents. So food is rewarding, make it well suited to serve as a mechanisms for reinforcing our social observations, our social interactions. But how? Let's look at the basic algorithms related to social and eating behaviors. So eating food to reinforce actions that lead to more food. And social behaviors involves other individuals' influence actions of oneself, which then feed back to the interactions with the others. Notably, it's not totally clear what is the mechanisms for this social reinforcing this social link. It's just like social contact, social touch, or could be other like the other primary rewards like food, right? So social context eating or eating during social context may clarify this by further reinforcing the actions that lead to more food, but also potentially reinforcing the social link. Okay, so I want to underscore because for most mammals, the social observation may be the first experience for us to learn how to the eating, maybe the first experience for us to learn the social observations. So this make it very fundamental that could be important for the other social cognitive behaviors that using this social observation, including this empathy or compassion. Okay, so now I was talking about okay how we incorporate this social context into eating behaviors. So to do that, we have two animals. One is called demonstrator, or you can call it teacher. The other one is called student, or you can call it observers. So the teachers actually they can free access to the spot. If they go to the spot, they can get a reward. But for the observer, for the student, they can obtain the full reward only when the teacher is eating. For example, you may ask, okay, what about when teacher is not eating? Actually, this is the case when teacher is not eating. For the student, for the observers, even they go to the food spot, they want to get the reward. So it's unlike the lab settings. So which lab settings, you don't need to pay attention to the others. You don't need to have the hunger. You're also very close to each other. So unlike the lab settings, these tasks incorporate these natural pressures, including hungers of these two animals, and also they are socially separated, and also they have the need, the survival relevant values of the social informations. So under these natural pressures, this task could reflect how the social behaviors and eating interact with each other. Okay, and this is how it works. You can see the teacher is, demonstrator is, observer follows, and then get food. Teacher eats, observer follows, and get food. And wait, yeah, demonstrate eats, observer follows, and then get food. So you do see actually eating is guided by the social partner. But you may wonder, okay, how do we know this is not caused by chance, right? So to answer that question, I do want to mention there are multiple chances for the observers to access to the food spot. So first is this active reward bot, which demonstrate it first, and then observers clicks, and then they get reward. Okay, so this is active reward should guided by the social partners, should guided by the demonstrator or the teachers. So there also could be the other cases. For example, in one case I just mentioned, so observer leaks when demonstrator is not leaking. You can see actually they didn't get reward. It's unrewarded bots. And also it could be this passive reward bot, which observers leaks first, and then demonstrator come, and then just observer just surprisingly get the reward. So both these passive reward or these unrewarded bots could be socially irrelevant, more likely happen by chance. Okay, so we further use this analysis to address, okay, whether this is caused by chance or not. But briefly, I will just mention we introduced a shuffling strategy, which estimated the possibility of chance, which is non-social performance of this ratio of active reward bot, which as here you can see, this shuffle means the chance performance. And then we compare our real data, real animal data with this chance performance, and then see whether it deviates or not. We do definitely see very clearly animals' performance deviate from these top 95% of these chance possibilities. And further, I convert this into a metric called social reward index, which shows you how many standard deviations above the chance performance our animals perform at. And we can clearly see these socially guided eating behaviors increasingly increased across these 2 weeks of training sessions. So that do show actually mice could learn this social context eating across 2 weeks of training, then we ask, okay, what information could the animals that first, does the social context really matters? So to do that, actually, we call it a simulated demonstrator session, which actually will remove the social context. There's no social context exists, but we play the real demonstrated consumption to these non-social pumps. So I mean, the non-social component is still there, but the social context, we remove it. We found actually, if we have the social context, it can significantly improve these eating behaviors. Okay, and also what sensory modalities of this social context really matters. We found actually the vision is important because visual block actually could surprise the socially guided eating behaviors. We also found the social familiarities will improve the eating behaviors. So that indicating, okay, the social vision and familiarity are important for these socially guided eating behaviors. But still, we are not clear about how the animals really use the social visions to get these eating behaviors. So to answer that question, I apply kind of 3 different steps. First, we ask, okay, certain types of behavior strategies to understand whether how the animals use the social context to get eating behaviors. First, we ask, maybe the animals has better coupling this their behavior to the like demonstrators' behavior to the teacher's behavior, right? It also could be the observers, they reorganize their behavior motifs. For example, they may form certain types of behaviors, eventually help the animals to show this, to gather social information. And then the final question is maybe they directly use the social observation towards the others, and then eventually get use this social information to get eating behaviors. So I will tell you how I test these 3 different mechanisms. So first, we use these deep learning tools called sleep, checking the animal poses, and then we kind of do these correlation mappings of these demonstrators, different set of the features, and also with observers is a different set of features, and ask, okay, whether these different part of features, they have a certain correlation. A long story short, we do find actually, for example, here in these learners, especially before the eating behaviors, we can see the facing, the social facing, which means the student, the observer's social facing looks like pretty sensitive to the demonstrators' behaviors, indicating maybe a stronger behavioral couplings of these social facings during this food seekings in these observers. So you do see some indication of the behavioral couplings in these social context eating. But what kind of these behaviors is, we further use another strategy, which we still use the sleep, these deep learning tools to checking the observer's behavior features. And then we apply unsupervised learning to ask, okay, what kind of behavior motifs they have. Okay, so we do find actually there are 12 different types of behavior motifs we can identify. And the important part is compared to the early stages with the later learning stages, you do find some new motifs formed. This is actually, we check, compare with the videos, these 3 different motifs. This very related to these head orientations, means they become more adept to turn around they head orientations during later stages. So, and then, so the conclusion of this part, we do find actually mice show these emergent head orientation motifs. And then we ask, okay, is this head orientation really related to like social observation or not? So to do that, actually, I analyze the social observations. I develop a software called observational editors, so that we can very accurately label these social observations. And then I develop a machine learning tools called XGBoost classifier, and then to infer these social observation events of our observers. And actually, these tools achieve similar performance as compared to our human validation, so that we can analyze hundreds, millions of frames in our videos across these 2 weeks of training. And we do find actually the mouse for our like student or observers, they do form certain types of policies which they should increase the observational rate towards the others across these 2 weeks of training. And important thing is we found this social guided eating directly increased with this social observation rate. So this indicating they could refine these social observations to get eating behaviors. Okay, so that indicating the behavioral mechanisms for using these social informations to get eating behaviors during the need interactions. But what is the mechanisms? When we consider this a mechanism during such need interactions, actually there's little know about it. We know a lot about like separate needs or motivation, for example, need for food, need for social, but the biology of need interaction is largely unclear, understudied. So let's just take a look at the circuit mechanisms of the social or of the food neural pathways. So we found actually, although they have very separate like food pathways and social pathways in terms of the sensory processing or the action processing, but one component, for example, these reward circuits do use both important, for example, VTA dopamine do use as a conversion point for the social and food motivation. So our simple question is, maybe the reward circuits may be involved for these need interactions, right? So to understand that questions, and then we directly monitor the VTA dopamine, we do find actually VTA dopamine may play the roles. So here's the evidence, which found actually VTA dopamine do encode food informations for self-eating, as I actually my previous work revealed, but also the VTA dopamine activate during other start to eating. When the teacher start to eating, you can also see the VTA dopamine showing the increased activities. So it means they also encoding the food information of the others. So further, we found actually they encode more complex social informations, including the encoding where is the teacher is, and also what posture size of the teacher showing the postures, and also how close the teacher close to the student, to the demonstrators. And that is suggesting, okay, the VTA dopamine do encoding these complex social informations and maybe use this rich social information to get eating behaviors. So this hypothesis is actually further supported by evidence we show this is a result in learners. But in non-learners, actually, we found the VTA dominance didn't show this relationship with the VTA dominance with this like spatial or postural, these proximity features of our teachers. So that indicating the social information transfer to may really regulated by the VTA dominance. But still, correlation doesn't mean causations. What about the causal roles? So to understand, to answer that question, I directly supply the VTA dominance when we like when the observers are observing the others, and then we do find actually if we surprising these VTA dominance, the social information can be less able to get these social context eating behaviors. So that directly suggests, okay, VTA dominance will support such eating interactions. Okay, so we also found bigger implications of our work. So traditionally in the field, in the social field, people think about it's a social interaction, it's a social context, social contact directly reinforcing the social motivation. So my question is, could this primary reward, for example, this food reward, could reinforcing these social motivations? So to answer that question, I directly test, for example, if the mice have these social context eating experiments, the food reward may improve these social cognitive behaviors, even empathy and compassion, right? So to test that idea, actually, the mice have these 2 weeks of social context eating, and then exposed to, for example, here paradigm is social empathy fear, and then I compare the social group versus non-social context eating group. We do find actually they show increase of social observation. Actually, data I haven't put here, they also showing higher social empathy feel. And also in another case, they also showing this increase of social elevation of food neophobia. So you do see actually this social context eating improve the social cognitive behaviors, especially the behaviors related to empathy fear, right? So the big implication is the primary rewards may reinforcing these higher level motivations. That maybe explains the evolutionary origins of the empathy and the compassion. So a short summary of this part of work. So I developed a task called social context eating task, reflecting how social needs and motivations interact with food motivations. And we do find actually mice can use the social visions to get eating behaviors. And then we found mice develop a social observation strategies. And we found VTA dopamine ions could support such eating actions. And also we found this food reward, primary reward, reinforcing these social motivations, especially these empathy-related behaviors. Okay, so and also this also open up many other questions related to the empathy and the compassions. For example, because we know the empathy compassions is not sometimes not just for the negative part, but also for the positive part. So how this opposite social information is integrated to the motivation systems. So my preliminary results showing actually there could be two pathways for getting this social information from the hypothalamus to the VTA system. One is activate the VTA system. One is suppress the VTA systems. So right now I'm testing this hypothesis and also how the other system, neural systems, could promote the need interactions. Right now, I do have some preliminary data showing the oxytocin systems in our hypothalamus could also promote such need interactions, and also how the social visions could using for sensing the other's needs. So I'm also testing another pathways from midbrain to the other part of the midbrain may support such systems. And also the final questions, what is the general computation rules for encoding for such need detection, especially related to empathy and compassion? So yeah, now I was talking about the last part. So talking about how my ongoing tool development to understand the computation rules. So actually, there's a very appealing hypothesis called the neuromodulator hypothesis, which means distinct needs or motivations are encoded by distinct neuromodulatory signalings. So this hypothesis is appealing because these neuromodulatory signals, for example, neuropeptide release, they actually can have very different timescales to encode different types of needs or motivated behaviors. And also, these neuromodulatory signalings can come from different part of the brain or come from even different part of the brain, or different part of the organs, body parts, make it well suited to encode different like needs, different motivations, right? But to test this hypothesis is quite challenging. This is because there are over 100 genes to encoding these different neuromodulator signals or its receptors. So the current strategy in the current field is only links one cell type at a time to certain behaviors, make it really hard to test this hypothesis at such scale, right? So our lab actually 6 years ago developed tools called gamma imaging. We can do the in vivo imaging to capture the behavior specific neurons, for example, hunger neurons, drinking neurons, and then we can do the ex vivo spatial transcriptomics to get the molecular expressions of the same neurons that has been in vivo imaging before. So with that tools, we can directly link these different molecular informations with these system level functions. But the limitation of these current methods is we can only profile 12 genes in thin sections, making it really hard to test this hypothesis and also relate to this in vivo neurons to this ex vivo neural slices. So to overcome this challenging, also supported by Sanford Institute, so I'm developing tools called ASICFISH, which refers to expansion assisted sequential in situ hybridizations, which combine two technologies. One is the technology developed by our lab like 5 years ago, which can profiling genes in the sick tissues. and also combining another technology called SickFish, which can we can profiling over hundred of genes easily in a limited timing. And then because there's a new challenges, we need to barcoding and not decoding genes in sick tissues. I also developed a software called 3D fish software and a pipeline which enable us to decoding the genes. And also importantly, actually we have applied this 3D fish kind of methods into different part of the regions and achieve a large number of gene profiling, and also my two-photon imaging also indicating there are separate neural populations in a certain brain regions to encoding the social or food motivations. So I also refer because this platform can cover over 100 genes, can cover different neuromodulating signalings. I also refer this platform as neural map, neuromodulating mapping across behavioral phases. So this platform will enable my lab directly testing this neuromodulating hypothesis of encoding different types of needs, especially the needs related to social and even related to empathy and compassion. So with that, I want to summarize. So my research studies the biology of needs and motivations. I propose three important frameworks to study it, including the need detection, the types of needs, and behavioral phases. And my past research has already identified many important circuits and molecules that related to physiological needs, safety needs, and social needs. And my ongoing work, especially supported by Sanford Institute, including this new behavior paradigms, new behavior pathways, and also with these molecular and computation tools, especially, for example, these neural map platforms. This will enable my research ranges the molecular and system neuroscience eventually to better understand the biology of basic needs and motivations, and especially the relevance to the empathy, compassion in animals and in humans. Okay, so with that, I want to thanks all the funding resources from the Stanford Institute, and also I really appreciate all the helps, collaborations within the Stenson group, and also my PhD mentor, all our collaborations with other advisors or mentors, and also these local San Diego communities. So with that, I want to thank you all for your attention, and happy to take any questions.